Hi, I'm Luke Livingston.
I'm Ian Fleming,
I'm Nihal Pandra.
and we're in period 5. The overall purpose of this experiment is to test the RNAi system in C. elegans.
Using sterile procedures, we gloved and goggled up, and then we bleached down our countertop. Then we got our plate of worms and our plate of bacteria.
Then we carefully opened up the bacteria plate with the double-stranded RNA and bleached the spot where we were going to place the adult worms so that the mutations would start with the new generation. We then opened up the plate with the worms and used an inoculation tool to scoop the worms off of the plate.
We then placed those worms onto the bleach in the bacteria plate. During this time, we closed the lid as soon as possible to limit the amount of other bacteria or other dust getting into the plate. We also kept the lid away from our faces and placed it over the Petri plate.
In this experiment, our model organism was C. elegans. C. elegans move in an S-like motion and are semi-translucent with an earthworm-looking body.
Also, C. elegans are a type of roundworm. They eat bacteria and they live in soil. C. elegans have a 3 to 4 day life cycle. They're hermaphroditic, and the females can lay from 250 to 1,000 eggs. C. elegans start off as an egg, and then they slowly hatch and become L1 worms, which are the smallest type of worm.
From the L1 stage, they move on to the L2 stage, and then from the L2 stage, they move on to the L3 stage.
From the L3 stage, our worms grow into L4s, and after, they grow into adults. In the bottom right-hand corner, you'll see an egg, where the C. elegans begins its lifelong journey as it grows into an adult.
As you could see right here, this is an L1, and you could tell it's an L1 because it's the smallest worm outside of an egg.
Beneath the arrow, you'll see an L2. The L2 is slightly smaller than an L3, but also slightly larger than an L1. In the top left of the screen is an L3. You can tell it's an L3 because it's smaller than an L4, but bigger than an L2.
You could see an L4. You could tell it's an L4 because it's slightly smaller than an adult, and it doesn't have any eggs.
In the upper left-hand corner is an adult. You can tell it's an adult because it's bigger than all the other worms, and it also has eggs in it.
The gene that was turned off in this worm obviously had something to do with the sporadic movement and seizures inside of the worm's body. In this mutation, it seems to happen early on in the life cycle. The gene that was turned off in this worm obviously had something to do with the shape and the size of the body of the worm. This gene mutation seems to show itself at around the L4 or adult stage.
The gene that could have been turned off in this worm could have had something to do with them causing some blisters on the skin. And in this mutation, it seems to show up towards the later stages around the adult stage.
The RNAi pathway in this experiment was used. The first step, double-stranded RNA, was produced in the bacteria and eaten by the C. elegans. The dicer inside the worm cells chopped up the double-stranded RNA into siRNA. The third step, the siRNA, then binded to several proteins to create the RISC.
The double-stranded siRNA are unzipped, leaving a single strand of RNA. Then RISC probes the cell for mRNA that it recognizes.
The RISC then binds to the matching mRNA and cleaves it. Then the mRNA can no longer be translated into a protein. Therefore, the gene has effectively been turned down or silenced.
Personally, I enjoyed this experiment because I thought it was fun looking at C. elegans under the microscope, and what I personally learned from this experience was how to use sterile procedures of transporting worms from plate to plate.
I really enjoyed this experiment and learning all about the C. elegans and the RNAi pathway. I'll definitely take apart and study the RNA pathway much more. It's really interesting and cool to me. I definitely took that from this project.
I really enjoyed this experiment because we got to look at the C. elegans under the microscope. I thought that was really cool. And I also learned about how to transfer worms from plate to plate and how to use the RNAi system to create gene mutations in the C. elegans.